Hello everyone. In this lecture, I am going to discuss about complementary DNA. Complementary DNA is DNA synthesized from a single-stranded RNA, example messenger RNA, using an enzyme reverse transcriptase. As we have already studied, when RNA is synthesized from DNA, that process is called transcription. Similar way, when DNA is synthesized from single-stranded RNA or RNA, then it can be called as reverse transcription process. Now, reverse transcription process is fulfilled by using an enzyme which is called reverse transcriptase. In this lecture, we are specially going to focus on complementary DNA synthesis. There are mainly 5 steps involved in complementary DNA synthesis: isolation of messenger RNA, reverse transcription, oligodecetailing, alkali hydrolysis, and addition of oligo-DG primer. This is the diagram of copy DNA synthesis or complementary DNA synthesis. Always remember, messenger RNA of eukaryotes contain a long polyadenylated tail at 3' end of their strand. Now, second step is reverse transcription step. In reverse transcription step, mRNA strands are treated with oligo-DT primer, reverse transcriptase enzyme, and 4 deoxynucleotides: DATP, DCTP, DGTP, and DTTP. DATP is deoxyadenosine triphosphate, DCTP is deoxycytidine triphosphate, DGTP is deoxyguanidine triphosphate, and DTTP is deoxythymidine triphosphate. Okay, so now what will happen due to this? Reverse transcriptase will add this deoxynucleotide towards 3' end of copy DNA, and this oligo-DT primer will bind to adenine. As we have already studied in DNA structure that adenine will always make a pair with thymine. Now step number 3, which is oligodecetailing. This DNA RNA hybrid is treated with enzyme terminal transferase and nucleotide DCTP. Now terminal transferase will add this DCTP or DC primer towards 3' end of mRNA strand and copy DNA strand. You can see oligoprimers in both the side, right? Now what will happen? The residues of cytosine which are on the copy DNA 3' end or 3' end will get curved, or they will form a hairpin loop like structure. Now step number 4 is alkali hydrolysis. Now due to alkali hydrolysis, what will happen? By use of alkaline sucrose solution, the mRNA and copy DNA strands will get separated into a single strand. Now there is only one strand. Now last and final step. In final step, it is addition of oligo-DG primer. What will happen? Oligo-DG primer is there and it will bind to this cytosine. Now cDNA here will act as a template for synthesis of double-stranded DNA in presence of enzyme DNA polymerase and deoxynucleotide precursors, which you can see over here. DNA polymerase 1 will add all these deoxynucleotides one by one towards 3' end or site of the primer. Okay, so double-stranded DNA formation will take place. Now, due to this, a single double-stranded DNA clone is synthesized, the length of which is similar with the length of mRNA which was utilized before. So in this way, from one complementary DNA or single strand of complementary DNA, double strand DNA can be obtained. And here S1 nuclease enzyme is used to open the hairpin loop structures which are generated during synthesis of double-stranded complementary DNA. I hope you all have understand this procedure. Thank you for listening to this lecture.